And so here we see the liger. This is Hercules, one of the largest cats that we've ever seen. And so he's a cross between a tiger or a lion and a tiger. He's the liger example. Here we see the Jaglion. Right, and so just a really interesting looking cat. And then here we have the Zonkey. And so this is a donkey and a zebra cross. And guys, while all of these crosses are possible, I want you to think about why we don't see more ligers running around in the open, or zonkies. Think about distribution of these animals. Typically, we're going to have more than one of these isolation mechanisms working together. And so generally speaking, think about lions and tigers, for example. Lions are found in Africa, associated with savanna type habitats. Tigers are associated with jungles of Asia and Eastern Europe. And so whenever we look at the different habitats and the regions that we find these animals, what we see is that, okay, tigers are way over here, lions are over here. We have a lot of geographic isolation that's going to keep them separated. However, in zoos and in different breeding institutions, we lose those geographic barriers. And so now individuals can come together, but even when they do come together, they end up producing an organism that is sterile. And so we have a pre-zygotic isolation mechanism working, and even if they do come together, we have a post-zygotic isolation mechanism in their sterility that's going to keep that particular hybrid from resulting into the formation of a new species. Right? Additionally, even though we do see a hybrid produced, and in some cases those hybrids may be fertile, what we see is that their next generation, so any cubs that might be produced by this individual, if he could produce them, and he can't, his sperm lack flagella, I believe it is, but even if this individual could produce a next generation, that generation would most likely be inviable. Remember that hybrid inviability we just talked about? And so we would see that they weren't really adept and couldn't deal with the stresses of surviving, and they'd probably all die off. All right, so we talked about those two. Now the last one is hybrid breakdown. Again, what we just talked about with the ligers, over time they become weaker. And we see this a lot, particularly in plants. You'll have a generation that's produced, and if you've done any farming at home trying to save seeds, or gardening even, you save seeds from one generation, and the next generation you plant them, they do okay, but not as good as the first generation. Well, you plant seeds from that. Third generation, you get nothing that germinates. And so over time, those hybrids become weaker and weaker. And that's an interesting way that different breeding companies control the seeds that they send out, which forces you to keep going back and buying more. And so over time, the hybrids get weaker and weaker. Okay, guys, now we'll pick up with the protists. Specifically, we're going to be focusing on the group called the protozoans, or what are known as the before animals. And so when we look at the protists, we see that this group appears about 1.5 billion years ago in the fossil record. So we have a good long historical record of them and their existence. But they most likely arose from an Archean ancestral group. And so their phylogeny tells us that animals and protists are most closely related to Archeans, those extremophiles that we looked at last class period. When we look at the protists today, there are 6 supergroups that are identified and have been classified. And so just to give you some background, up until the 70s, Kingdom Protista, as it was known, was basically a garbage heap for anything that didn't clearly fit as an animal, plant, or fungus. And so over time, you ended up having this massive accumulation of of organisms that were very different from one another in many cases, but also shared some similarities, generally, that they were single-celled. Now, as our technology improved, so is our understanding of what protists actually are. And so recently, beginning about the 90s, there's been a big push to clarify the phylogeny of these different organisms. And so this group is constantly undergoing rearrangement. They're constantly being moved and reclassified and reorganized based on new information that surfaces as we do more research. And so just bear in mind, the classification we learn today in 10 years may be void, and that's because we're learning more and we're working to better understand these organisms specifically. However, we're still going to talk about them. We're going to focus on 6 supergroups that are found in this protist category. What we see is that we'll have many that are autotrophic and some that are heterotrophic. And so this is one feature that we're going to use to differentiate them. And so the protozoans are the ones we're going to focus on. These will be heterotrophic organisms. So proto means first or before. Zoa refers to animal. These are the before animals. These are unicellular eukaryotes. So they all have cells with a nucleus. They're found in moist environments, in almost every type of soil or freshwater, seawater habitat. They're found in the deep sea, in the Arctic, and in the Antarctic. So they're very prolific. There are several characteristics that identify and are unique to the protozoans, and the first is called the pellicil. And so the pellicil is one type of specialized organelle we find in the protozoans. This is a regular arrangement of microtubules beneath the plasma membrane that are going to help maintain the shape of the protist, but also flex to allow for locomotion. We see that they will also have a contractile vacuole, and this is something that's going to help them maintain their water balance so that they don't burst. Essentially, it works kind of like a sponge. Water that's coming into the cell, because water moves from high concentration outside to low concentration inside the organism, water that's coming in through osmosis has to go somewhere. They concentrate it in this contractile vacuole. As the vacuole expands, it gets to a critical point, and then they squish it out or wring it out like you would a sponge or a wash rag. Once it's wrung out, all the water is pushed to outside the body, then it can slowly fill up again, and they do it all over again. So it allows them to maintain their water balance, or essentially keep from bursting in their aquatic habitat. Now, we'll also identify various features associated with locomotion in the protist, and this is another point for differentiation. We see that some groups will move by pseudopodia. Pseudopodia are false feet. Pseudo for false, pod for foot. This type of locomotion is seen in the amoebas and their relatives. Other protists are going to move using a flagellum. The flagellum is a long whip-like tail that is covered by an extension of the plasma membrane, and it's going to wiggle back and forth, kind of like a fish's tail would, to push the cell through its medium. Lastly, we have cilia. Cilia are small, hair-like extensions, again covered by the plasma membrane, so they're part of the cell, but they're going to move kind of like oars on a boat, and so they'll row the animal, kind of like your little arms would, through the water, through whatever medium that they're in, and pull them through their environment. Now remember, we're looking at something in 2 dimensions here, so this is a cross-section through. These will cover the entire surface of the cell, or they may be in strips or bands along the surface of the cell, depending on what species we're looking at, but they're all going to look at these small hairs, or eyelashes. Now, whenever we look at metabolism in the protozoans, all metabolism is going to be heterotrophic, which means they're other feeders, or they're feeding on some other type of organism to get their carbon they need for organic molecules. We'll see many of them will be free-living individuals that are going to be feeding on other molecules or other organisms in their environment, such as this amoeba that's getting ready to take this bacterium in for a meal. In this, we'll see that they're just basically hunters or predators. We also have a number that are parasitic, and so we'll talk about parasitism here in a minute. And so others exist in a symbiosis, or a close association or relationship with another species. There are 3 types of symbioses that we will talk about this semester. The first is a mutualism, where both individuals benefit. And so here you have a water buffalo, and those are oxpecker birds that are getting a ride there. The oxpecker birds benefit in this relationship by getting a meal from the water buffalo. What they do is they will peck off any parasites, ectoparasites, that they come across. So things like ticks and fleas and basically flies that come by, or larvae that are present on the body of the water buffalo, they will clean them off. So it's kind of like a buffet for them. In exchange, the water buffalo is going to be rid of these disease-causing organisms, and so that's going to help him stay healthier. So both organisms are benefiting from this association. Now, here in the center, we have a commensalism. In a commensalism, one member is going to benefit, the other is neither helped nor harmed. And so here you see cattle ears, these white birds, in this field with these cows. Now, these cows couldn't care less if those birds are there. They're walking along, grazing, doing their thing. As they walk, they move the grass, which stirs up the bugs, grasshoppers and things like that. The egrets benefit, the birds benefit, because that helps them locate food. Cattle, they're neither helped nor harmed. Okay. Lastly, we have a parasitism. In a parasitism, one member is going to benefit at the expense of the other. And so here you have a tomato hornworm, this caterpillar, and these are found in gardens. They produce a sphinx moth, which we'll talk about at some point. But you see, most of them don't have these little white things all over their backside. Those are pupating wasps. And so what we see is that braconid wasp, a specific type of wasp, the female will pierce the body of the hornworm, lay her eggs inside of him. Those eggs hatch, and you get these tiny little larvae that kind of cruise around through caterpillar guts, eating their way through him. And the caterpillar can't really do anything about it, and so he's just kind of walking along, trying to eat, trying to survive. Well, eventually he gets so lethargic and just no energy, because he's literally being eaten from the inside out, that he just stops moving. At the point of maturity, the little larva will actually eat a hole in the body wall, and they will pupate in these little cocoons on the exterior portion of the hornworm. After they finish pupating, they will emerge from their pupa and fly off to begin their next life cycle as an adult. And so parasitism, the braconid wasp benefits. The hornworm obviously is the one that is going to die, and is paying for this relationship. Now, keep in mind, not all parasites kill their host, because they're losing their food source if they do that. Many of them will live for years inside or in association with their host before there are any detrimental effects or effects severe enough to cause the organism to die or the host to die. Now, remember that a whole group of protists are autotrophic, and they're going to be the algae. We're not going to talk about those because those are going to be more in association with the plant kingdom. But there's a lot more out there than just the protists, the protozoans, that we'll be focusing on today. All right. So here's the summary of the taxonomy that you're responsible for. So for protozoans, we're going to be in domain eukarya up here at the top. The group, we just generally informally call these the protists. These are all going to be single-celled eukaryotes. And there's 4 supergroups that we're going to be talking about. We're going to be talking about excavata, amoebozoa, rhizaria, and alveolata. Right? And so again, even though this supergroup classification is outside of those 8 hierarchy taxa that we talked about with Linnaeus, supergroup is still applicable. So just keep that in mind, that that's something that's just a little bit different, and that's because the protists are just a little bit different. Okay. All right, so we'll begin with supergroup excavata. This group, commonly called the excavates, are named for a feeding group that many of their individuals possess. Right? So supergroup excavata get their names from their feeding group, and so you can see one right here. You can see this feeding group in here as well, and so that's where they get that name. They're all going to move via 1 or more flagella, and so here you see this is the euglena with its flagella illustrated. Here you have a flagella sticking out at this end. This one has 2 flagellum, and you can see some individuals in this group with hundreds of flagella. They're all going to have that pellicule that we talked about, and I love this picture here in the green because you can see those microtubules beneath the plasma membrane giving this organism its kind of textured, almost looks like a fingerprint. Those are the microtubules of the pellicule that are going to help keep its shape but allow it to move. It's still flexible, and you can see it here on the euglena diagram as well. This group is going to be defined by the presence of only a single nucleus. There is one group that we'll look at that is multinucleated, and so those end up looking a lot like the excavates, but this is one important feature that distinguishes them. Our representative